Dad, why are we going to the zoo today?
Well, Timmy, I thought it'd be a great idea for us to visit the zoo and learn about different animals and their unique attributes.
I can't wait. I love going to the zoo. Can we go see the lions first?
Of course, Timmy. Let's go to the viewing area. Lions are truly amazing creatures.
Dad, look at its fur. It's so different from the other animals.
You're right, Timmy. Lions have a very unique fur coat.
Look, there's a sign for the Worlds of Colors exhibit. Can we go there? I want to see some animals with very unique colors.
Absolutely, Timmy. The exhibit is all about the range of colors in the animal kingdom. Let's see what we explore and see what we discover.
Look, Dad, parrots with so many colors. They look like rainbows.
Nature is truly an amazing artist. Each animal has its own unique palette.
Dad, what's melanistic?
Melanism is a special coloration where animals have an abundance of dark pigment called melanin, which is the opposite of albinism. Let's see what creatures here have melanism.
These jaguars have different colors than the ones on the left. Why are they so dark, and do they share similar characteristics?
These animals are so dark due to genetic variation affecting the production of melanin. In melanistic animals, there is an increased production of eumelanin. Both types of jaguars share similar behaviors. However, melanistic jaguars have camouflage in certain environments.
So it's because of something in their genes that make them so dark?
Yes, Timmy. The melanocortin-1 receptor gene plays a crucial role in regulating the production and distribution of melanin. The pigment is responsible for the coloration of the skin and other tissues. I hope you enjoyed your time at the zoo, Timmy. How about we go home and we can research more about melanism? This can maybe even help you with your science fair project.
Can't wait. Now that we are home, can we research more about why some animals like jaguars have melanism?
Yes, Timmy. First, let's see how this mutation process all starts. First, we must understand what a mutation is. A mutation is a change in DNA that changes genetic information. The change in DNA causes a change in protein, which results in a completely different trait.
Oh, okay, that makes sense. Mutations must be pretty bad then.
Well, not all the time, Timmy.
What do you mean?
For example, some people are able to get disease immunity from mutation, which is actually a good mutation. In other cases, like developing a birthmark, is a neutral mutation. It has no positive or negative effect. But different types of disorders can be results of mutations, which is not very good.
Oh, okay, that makes sense.
So the overview of this process is that DNA transcribes itself onto RNA. RNA is then translated to create amino acids, which code for proteins. Finally, these proteins are what code for the traits.
Dad, this is kind of confusing.
Don't worry, Timmy. I'll go into detail of each step. It all starts with something called enzymes. They're like chefs in the kitchen, cooking up the pigment, melanin.
Enzymes are master chefs, but why melanin?
Like I said before, the melanin gives animals their color, and to make the recipe, it involves a special gene. Let's find out what it's called. So it looks like the MC1R gene is responsible for this. If there is a mutation in this gene, it's like changing the chef's recipe card.
So mutation in the MC1R gene leads to a different pigment recipe? Got it.
Next, we have to understand what DNA is. DNA is like the master copy of all the nitrogenous bases. Think about it like a thing that gets copied by the mRNA. Now, I'll show you why mRNA is used. Let's search this up as well. Okay, so it looks like the mRNA is like the chef's assistant who copies down the recipe from the DNA and brings it to the kitchen where the action happens.
mRNA is a messenger delivering the recipe to the kitchen? That makes sense.
And guess what this kitchen is? It's a tiny factory called the cell. Imagine a kitchen inside the cell, working day and night.
The cell is in the kitchen, but how do they follow the recipe exactly?
Next, I'll have to explain to you a process called transcription. Let's find a picture so I can explain it better. Timmy, check this out. I made a visual guide to the transcription process.
Wow, that's impressive. Let's look at this picture together.
Okay, first let's check out all the enzymes in this picture. There's RNA polymerase, which synthesizes RNA molecules from a template strand of DNA. And then we have helicase, which separates the double-stranded DNA by breaking the hydrogen bonds into single strands, allowing each strand to be copied. The RNA is used in this process because it acts like our recipe card. Where do you think all of this happens?
Let me guess, the nucleus?
Yes, good job, Timmy. Now this is where complementary base pairing comes in. It's like a matching puzzle. Adenine pairs with thymine, and cytosine pairs with guanine. And in RNA, thymine is replaced with uracil. This complementary base pairing ensures the recipe is copied accurately.
Like fitting puzzle pieces together. Interesting.
Next, you have to understand 3 different types of RNA. mRNA carries genetic information from the DNA in the cell nucleus to the ribosomes in the cytoplasm. The information encoded in mRNA is used by ribosomes to synthesize proteins through a process called translation. rRNA is a structural component of ribosomes, which are cellular structures responsible for protein synthesis. Along with proteins, rRNA helps form the ribosomes and provides a platform for mRNA and tRNA to interact during protein synthesis. tRNA is responsible for bringing amino acids to the ribosomes during protein synthesis. Each type of tRNA is specific to a particular amino acid, and it carries the appropriate amino acid to the ribosome based on the information encoded in the mRNA.
So tRNA, mRNA, and rRNA are the team making sure the recipe is perfect.
Exactly. Now we have to understand what codons are. So codons are a sequence of 3 nucleotides, or triplets, in mRNA that encode for a specific amino acid or start or stop protein synthesis. The sequence of codons in mRNA provide the information necessary for the assembly of amino acids into a polypeptide chain during the process of translation. For example, the triplet AUG is known as the start codon, which always signals the start of protein synthesis, and UAA, UAG, and UGA are all stop codons, indicating the stop of protein synthesis. Next, we have anticodons. Anticodons are a sequence of 3 nucleotides located on transfer RNA molecules. These anticodons are complementary to codons present in mRNA. During protein synthesis, tRNA molecules play a crucial role in bringing the correct amino acids to the ribosome, based on the information encoded in the mRNA. Next up, we have amino acids. Amino acids are organic compounds that serve as the building blocks of protein. They play a crucial role in various biological processes, including the synthesis of proteins, which are essential for the structure and function of the cells. There are over 20 different amino acids commonly found in proteins, and each has a unique structure and function. Now that we know all this, we can talk about how a protein is assembled. This process begins with the initiation of protein synthesis. The mRNA molecule binds to the small ribosome subunit. The tRNA that is carrying the amino acid binds to the start codon on the mRNA. The start codon is AUG. Then, the ribosome reads the mRNA sequence, and the tRNA molecules bring the corresponding amino acids to the ribosome. Peptide bonds are then formed between adjacent amino acids. After the formation of each peptide bond, the ribosome moves along the mRNA to read the next codon. This movement is also known as translocation. The process continues until a stop codon is reached, UAA, UAG, or UGA on the mRNA.
Codons and anticodons are like secret messages for chefs. Nice analogy.
Next, we have to look into translation, which is our last step of our adventure. Let's find a picture online.
Okay.
Oh, this diagram looks pretty cool. Now, since we already know what every type of RNA does, let's learn what happens during this process of translation. Now, imagine building a protein chain by assembling a delicious dish. It starts off with the first amino acid joining the chain. In order to finally assemble the protein, it's like building a chain with peptide bonds. Think of it as linking sausages together. And when the chain is complete, the protein is ready to serve.
A protein chain just like linking sausages. That's easy to picture.
Now let's look at mutations. You can think of a mutation as a typo in the recipe card. Let's see what causes melanism. So it looks like melanism is caused by a point mutation, where a letter in the recipe got swapped, a missense. Let me show you with this visual. As you can see, since one of the nitrogenous bases was switched, it resulted in a completely different protein being created.
Oh, so it's like a typo in the recipe card changing the dish?
Yes, and when these typos happen, it can change the entire dish. The melanistic jaguar that we saw earlier are the special result of such a weird genetic typo. Some mutations might not be noticeable, but mutation such as melanism is very visible.
Wow, that's a lot of information. Is there any other way to remember all this?
Think of it as a colorful kitchen, where the chefs, which in this case act like the enzymes, are following a detailed recipe card, or the RNA in the heart of the kitchen, the nucleus. The ingredients, which act like the complementary base pairs, are matches, creating a masterpiece, our protein dish.
Dad, thanks for helping me understand. Let's go to the zoo again tomorrow.
Sounds like a plan to me. I'm really proud of you.